November 5th. And now she's 60, riding down the highway with her friends at her side. Her life without them wouldn't be complete.
Hello, my thickness. It's your host, Fendi Williams, and today on my show, we're going to be talking about Kim Kardashian and her new baby, Chicago. But first, we have a guest. He's a scientist. His name is Dr. Kim Kardashian. He'll be telling us some of his research he has recently done. So, can you all give him a big welcome, Mr. Kardashian? Hello, doctor. Hello. How are you doing?
I'm doing fine.
So, tell me some of the stuff you've been doing.
So lately, I've been doing Botox, lip fillers, injection.
No, not that. About your research on regeneration.
Oh, sorry, I find it very important for me to discover and research new ideas to help the human population. Over the past few years, I've researched about regeneration of tissues in order for me to discover if humans would be able to regenerate limbs, and I was able to. I was able to do this by extracting crucial genes that made it possible for other organisms such as salamanders and axolotl. With the extracted genes, I injected them into a human cell, hoping that it would attach onto a gene. Once that cell would divide, the DNA would hopefully split equally, now leaving us with two cells carrying the desired gene. Two genes that are required for self-renewal of germ cells in mice and flies, Pwi-like 1, PL1, and Pwi-like 2, PL2, are expressed in limb blastmel cells, the basal layer keratinocytes, and the thickened apical epithelial cap in FGF8 is transcriptionally downregulated in the presence of the morpholino oligos, indicating PL1 and PL2 might participate in FGF signaling during limb regeneration. Given the requirement for FGF signaling in limb development and regeneration, the results suggest that PL1 and PL2 function to establish a unique germline-like state that is associated with successful regeneration.
So, how do you activate these genes?
Well, I activate the genes by...
Regeneration involves mechanisms similar to maintaining embryonic stem cells. They are a great step towards regeneration of new tissue because developing embryos have a high growth, self-renewal capacity and ability to differentiate into some precursor cell types when directed by the appropriate environmental factors. We also see embryonic stem cells creating and growing to make limbs as the baby is developing, so their ability to do so is already evident. However, we do see regeneration in adult stem cells regenerating epithelia, brain tissue, muscle, blood and bone. We have decided to reprogram adult cells to revert to ES, or pluripotent stem cells. Pluripotent stem cells are cells that can give rise to any type of cell in the body except those needed to support and develop a fetus in the womb. With the reprogramming of the cells, they can de-differentiation. The de-differentiated cells can then differentiate to reconstitute the lost tissues at the site of the injury. Regeneration starts with the formation of blastema, the composition of which has long been thought to consist of undifferentiated pluripotent cells derived through the process of de-differentiation is the reacquisition of embryonic-like developmental potential of the blastmel cells. Blastema is the reactivation of embryonic genes recapitulating the events that occurred during embryonic development. The formation of the blastema itself represents a transition phase in which limb cells respond to injury by differentiating to become embryonic limb progenitor cells that can undergo during this phase. Rapid wound closure is followed by the de-differentiation of limb cells to form the blastema. Nerve axons in the apical epidermal cap (AEC) are both essential for the formation of an accumulation blastema. Regenerating axons induce the synthesis and secretion of the anterior gradient protein, AGP, by distal Schwann cells during de-differentiation and by the gland cells of the AEC during blastema growth stages. AGP promotes the regeneration of a denervated limb. 2-digit stages when into the limb during de-differentiation. Once a critical mass of blastema cells has been attained, the blastema can undergo morphogenesis in the absence of the nerve, but the regenerate will be a miniature, because the nerve is no longer inducing the AC to carry out its AGP-mediated proliferative function. The stages remain dependent on the nerve for blastema growth. Keep the nerve and in a couple of months and limb will grow to its full size.
Oh, hi, Dr. Kardashian. Thank you for coming.
Thank you for having me.
Thank you.
Goodbye.
So, our next guest will be a patient that Dr. Kardashian has actually implanted this new regeneration gene into. Everyone give a warm welcome to Fernando.
Hey, how are you?
Yeah, thank you for coming. So, Fernando, why did you want this regeneration gene implanted into you?
Well, I mean, it helps with the lumberjack industry. And finally eat tacos, not worried about injury.
During lumberjacking?
It's tough business. Yeah. A lot of wood. A lot of accidents.
So limbs get cut off.
All the time. In fact, actually, my limb just got cut off a couple weeks ago. So, I got a baby arm.
So have you had any accidents since the procedure of adding the regeneration gene?
Yeah, in the lumberjack industry, it's kind of common. A few months ago, like right after the procedure, I tested it with a chainsaw of mine, so no accident at all, just full stupidity, but I needed to see what Dr. Kardashian was on about. So, yeah, the pain is all still there. I almost fainted, but luckily the cells decided to like stop bleeding and it covered over with a blastema.
Alright, yeah.
Right. So I have no idea what that is, but Dr. Kardashian told me that's what it was.
Okay.
Right?
Okay.
And I went straight to the emergency room after the doctor was there with me the whole time, and without Kardashian explained to me that my baby arm at the time, it was actually a baby arm. It was just
like
it was just
full
my arm.
baby.
Full baby, like imagine a baby arm on an adult body, that was it.
Okay.
Right? And he was just explaining to me that that's actually my regenerative arm growing back, and it's taking a good minute. It took a couple of months for
Okay.
my full arm to grow back as it is now. As you see, it's not a baby arm anymore.
Okay.
And that was just the that was just the regenerative arm being reproduced by the blastema, and it was taking a while for the cells to decide what function it was going to take as my new arm.
Okay.
And it just, without that, I wouldn't have this arm again. Well, I mean, if I didn't chainsaw my own arm.
That's true.
But, you know,
Well, thanks, Fernando. Thank you.
yeah.
Everyone give a big hand to Fernando. Thank you for coming.
Yeah.
Thank you.
November 5th, and now she's 60, riding down the highway with her friends.
Next, we're going to talk about Kim Kardashian and her new baby, Chicago West.
They are her joy and pride. She's learned a lot from all her good friends.
Hi, my name is Carla Shaw. You can stop at 5 or 6 stores or just one. I don't need friends, they disappoint me.